Okay, in today's lecture, we're going to talk about the mitosis. So why we need the mitosis, why it's so important. So we will cover everything. So actually mitosis is one cell divides into two identical cells. So we're going to talk about it today. So each of the same number of the chromosomes as the original cell will have two identical cells. So let me share the screen now. So this is the mitosis and meiosis slide. Actually, we're going to only talk about the mitosis in this lecture. So definition of the mitosis and meiosis, we will talk about it. Then the mitosis, and we will talk about the cell cycle, and we will focus on the meiosis later. So actually, mitosis. So what it is? Mitosis is one of the one cell divides into two identical cells, each with the same number of the chromosome as the original cell. It keeps everything exactly the same. Okay, so chromosome number stays diploid. What's the meaning of diploid? Meaning of the diploid full set of chromosome. This is the overall number of the chromosomes in one cell. For example, the diploid number of the cell in human body is 46. So that means we have 46 chromosomes totally, okay? So mitosis is used for growth, okay, for the repair, and also it's used for the maintenance of the body. So we will talk about it. What's the maintenance? Maintenance means so you want to if you injured your body, so you're going to maintain it, you're going to fix it. In order to fix it, we need the mitosis in this case. So in case of the medical terminology, imagine a patient has a small skin wound. This is a small skin wound. To heal, nearby skin should divide by mitosis, always, to replace the damaged cells. They must be identical to the original cell, right? So you have one skin, and that skin, you damaging the skin, and from that skin should arise another skin. So they should be identical like previous one. In order to maintain this, we use the mitosis. They must be identical to the original skin cell to maintain its function. So you're starting the reproduction actually. So you started as a zygote, right? For example, the sperm cell, this is the sperm cell, this is haploid number, this is a half number of your chromosomes, and the egg is also half number of the chromosomes. They're joining together to make two sets of the chromosomes. We call that diploid number of the chromosomes. So you can see here the zygote. The zygote has diploid number, which is 46 chromosomes in the human body. So after the fertilization, the number of the the number of the chromosomes in the zygote will be 46. That zygote, after that, divides by mitosis. It's going to divide by the mitosis to make the embryo. So this is embryo. We got that embryo. We made that embryo by the division of the zygote by mitosis. Okay, so embryo then with the way of mitosis, after with the way of division of the mitosis, it's going to make the fetus. We call it embryo until 1 month of the pregnancy. We call it embryo. After 1 month, we call it fetus. Okay, and then it's going to be adult. We are going to grow, right? We're going to develop. So this is also by mitosis. All through will go through the mitosis as we age. Even as an adult, your cells divide via mitosis to heal the wounds, to grow your hair, to replace the red blood cells, all blood cells, and etc. Everything need mitosis. Okay. So what's meiosis then? So what is meiosis? So meiosis, this is totally different from the mitosis. Meiosis is one cell divides one cell, just divides to form four identically different cells. Sorry, four genetically different cells. We're going to make four genetically different cells from one cell. Okay, so cell is going to divide and make four genetically different cells, each with a half number of the chromosomes. Each is going to make half number of the chromosomes. Chromosome number becomes haploid, 23 chromosomes. For example, the sperm cell, from one spermatogonium, we're going to make four genetically different sperm cells with haploid in number. Okay, so this is N means haploid, 23 chromosomes. Okay, so in the egg of the females, they also have haploid number of the chromosomes, which is 23. Okay, so occurs only in testes. We call it spermatogenesis. So where the meiosis happens in the males, the meiosis only happens in the testes. Okay, we call it testes. In the females, it happens in the ovaries. Okay, in the ovaries only. So in the male infertility workup, if the sperm cells are deployed, imagine if the sperm cell is deployed, the patient may have the meiotic failure leading to infertility because the sperm cannot fertilize the egg. Okay, so sperm should be 23 chromosomes in number, not 46. If it's 46, the male is infertile. Okay, so in male cells in the testis do meiosis. Okay, the testis do meiosis, which makes 23 chromosome numbered sperm cell. In females, in the ovaries do meiosis to make the egg cell, which is 23 in number. The chromosome number will be 23. These we call the gametes. Okay, sperm cell and egg cell. Actually, we call it gametes or sex cells. Or sex cells. So the fertilization, actually, what's fertilization here illustrated? Fertilization, actually, when the sperm cell with 23 chromosomes is entering into the egg cell, which also has 23 chromosomes, and they're going to combine to make the zygote. So fusion of the gametes, fusion of the sperm and egg cell, this is called the fertilization process, okay? So then they create the zygote, which is 46 totals. This 23 is joining with this egg 23 chromosome. So they're fertilizing. After that, we're getting two N's. So N is combining with this N, and then we are making two N, which is 46 chromosome, and each N is 23 chromosomes total. Okay. So this zygote divides by mitosis, then forms the embryo, zygote, and adult. Okay, that's how the life cycle continues, as shown in this diagram. So mitosis is used for the growth, repair, and body cells, and meiosis is used only for the formation of the gametes, which are sex cells. And the chromosome number after the mitosis will be diploid. From diploid cell, you're going to obtain diploid cell in the mitosis. But in the meiosis, you're going to start with diploid cell, and then you're going to get the haploid cell. Number of the divisions in the mitosis is 1. Number of the divisions in the meiosis is 2. We divide the meiosis into 2. Meiosis 1 and meiosis 2. Number of the cells made in the mitosis, we make 2 identical daughter cells at the end. But in meiosis, we're going to make 4 non-identical gametes at the end. So where the mitosis is happening in whole body, all body somatic cells. Somatic cells, that means your body cells are going to do the mitosis. What about meiosis? Meiosis is only done in the testes by the sperm cells, and also it's done only in the ovaries of the females, which is happening in the egg cells. Okay. So mitosis is used for the skin healing. For example, after the surgical incision, the skin cells divide by the mitosis to repair the cut. Okay, the mitosis is also used for the red blood cell production. Bone marrow, we have a special structure which is called bone marrow, uses the mitosis to constantly repair the blood cells that die after 120 days. That means 4 months. So mitosis is also in the cancer cells, uncontrolled mitosis. When you cannot control the mitosis, that leads to the tumor growth. That's why the anti-cancer drugs try to block the mitosis always. Okay? If there is a problem in meiosis, there can be the Down syndrome. For example, if meiosis fails and the egg keeps 2 copies of the chromosome 21, if the chromosome which is in number 21 will have 2 copies, that means the baby gets trisomy. of 21 chromosome. That means the Down syndrome, okay? So in the in vitro fertilization, we call it echo in Russian, or IVF, in vitro fertilization in English. In vitro fertilization, the mature eggs, which are haploid in number, are collected from the ovaries, which resulted after the meiosis. Okay, so this is the explanation of that. So this is the chromosome number. This is the diploid number. Here you can see the diploid number. So out of the diploid number, these are the somatic cell. This is the somatic cell. This is the chromosome number of the somatic cell, which is body cell. This is somatic cell. So what's happening here? 22 pairs. We have 23 pairs of the chromosome, which is 46 total. This is 46 total. Out of that, 44 of them, or 22 pairs of them, they are called the aptosomes. Aptosomes means they are autosomes. Aptosomes means they are not sex chromosomes. Okay, so out of them aptosomes, and only one pair or two chromosomes at the end, they are the sex chromosome, which determines your sex. It can be XX or it can be XY. If it's XY, that means you're a male. If it's XX, that means the female body. So diploid number of the chromosome always in the human body is 46. If I divide it into two, I will get 23, which is haploid in number. So haploid means only one set of chromosome. For example, which came from the dad or which came from the mom. This is either if it's dads or moms. This is one set of chromosome. If they combine it together, if you have two of them together, now in each of your cell, you have the chromosomes which came from the dad and which came from the mom, and you have full set of chromosome, which is 46 normally. So that means diploid number, and out of that diploid number, which is 46, 44 of them are aptosomes and 2 of them are sex chromosomes. Okay, so if I divide that 46 into 2, I will get the haploid number, which I can see in the sperm cells or in the egg cells. We call it O. Okay, so here the illustration of the nematodes. In the nematodes, this kind of organism. It has 4 number, which is diploid. In order to find its haploid number, I just divide 4 into 2. So 2, it is nematodes haploid number. I hope you understood it. So this is also previous illustration, so I'm not going to stop on it. So we talked about it a lot. So their testes in the males and ovary in the females, they're going to make the sex cells, and each sex cell is haploid in number, which is 23 chromosomes in each. So when they combine together, when they fertilize, when they fuse, when the sperm fuses with the egg cell, they're going to make the zygote. These zygotes will have 2 and this diploid number of the chromosome, and it's going to make then embryo, which is also diploid number, and the baby will also grow diploid with the way of mitosis, which diploid number of the cell also divide. So at the end we age. Okay, so mitosis is really essential for our body. So let's talk about the chromosome. So chromosome, what is it? So what is chromosome? Why we are talking about 46s, 23s? So think of a chromosome like a tightly coiled thread of genetic instructions which contains the DNA. So inside of the chromosome, we have tightly coiled thread of the DNA molecules. So in each cell, in one cell, we have 2 meters of the DNA. In each of your cell, we have 2 meters of the DNA. And that DNA is tightly coiled inside of your chromosomes, which is 46 in number. So that means 2 meters are inside of 46 chromosomes. Okay, so chromosomes are the way where the DNAs are located, packed into the tiny X-shaped structures, which we call chromosome. Okay, just like in the image you can see here is X-shaped. Okay, so let's go step by step to understand it completely. So the DNA and chromatids and the chromosomes, let's talk about them. So DNA is a very long thread. It's very long. DNA is too long. That thread wraps around the proteins, which is called the histones, okay? Which is called in the histones. You can see. This is actually the DNA. As you can see, this is a DNA molecule. It's wrapped in the histones. You can see the histones. When the DNA is wrapped, that thread wraps around the histones. We call it chromatin. Okay, we call it chromatin. So chromatins, they are wrapped version of the DNA in the histone proteins. So they are the histone proteins. They're like in the wrapping. Imagine I have this kind of rope. This kind of rope should be wrap it in any object. For example, imagine this is the histone. I'm going to wrap it in the histone. Okay, if I wrap it, it will be tightly coiled. Okay, so histones are proteins which wrap the DNA on it to make the chromatids. During the cell division, chromatin coils up tightly, which make the chromosomes. Okay, those chromatins then they're going to coil up more tightly to make one chromosome. So a chromosome is just a super coiled DNA. Okay, so what's the meaning of coiling? This is the coiling. Okay, this is like wrapping. So chromosomes is just super coiled DNA which is ready to be divided and pass it on during the cell division. Okay, it's going to pass through the cell division. So let's talk about the parts of the chromosome. You can see the sister chromatids, right? So this is the one sister chromatid. This is another sister chromatid. First sister chromatid, second sister chromatid. Okay, first sister chromatid, second sister chromatid. So sister chromatids, when DNA is copied before the cell division, you get two identical strands joined together. Okay, this chromatid, this chromatid is going to be copied. So it's going to make this part, okay? This part is going to make the second part before the cell division in the S phase, okay? In the S phase of interphase. It's going to make the second part. So this second part actually is made during the S phase of the interphase. Okay, each of these strands we call then chromatids. This is one chromatid, second is new synthesized second chromatid. So that means they're totally generally called sister chromatids. Okay, the two chromatids together now called sister chromatids. They are identical like twin pages printed from the same file. Okay, they are identical because second one copied from the first one. That's why identical. So what's centromere? So centromere, which is in the center, it's the waist where the chromatids are connected in the center. It holds two chromatids together. You see the first chromatid, this is the second chromatid. So first and second chromatid, and uh, in the center, you can see the centromere. Okay, this centromere is actually holding two chromatids together. And the spindle fibers, we're going to talk it a little bit later. Spindle fibers attach here during the cell division to pull chromatids apart. Okay, so without the centromere, chromatids would float away randomly. If it's not holding two chromatids together, just sister chromatids together, that means they're going to float themselves. Okay, it's like the belt. You know what's belt? It's like a belt that holds them tightly, which is holding the two sister chromatids together. Okay, I hope it's understandable. So what are the spindle fibers? We have special structures which are called the centrioles. They're going to make the spindle fibers, and those spindle fibers are going to attach here. The chromosome has two arms. The first is called the P arm. The second is called the Q arm. Okay, so P arm, actually the short arm. P from the petite. From the French, it means small. Okay, P arm means small arm. The smaller arm is always called the P arm. The Q arm is a long arm. Okay, this is how chromosomes are mapped in genetics. For example, genes might be located on 7q31. Okay, that means imagine this is always used in the map. It's mapped in the genetics. If 7q and 31. If you see this kind of number, that means chromosome number 7. Q means this is the Q arm, which is long arm, and 31 means band 31. This is just, we are using it for mapping for the classification. Okay. So what about the telomeres? Let's focus on the telomeres. These are the end caps. They are end caps. They are end caps of the chromosome found in both tips. They're found in the both tips, made of repeating DNA sequences. They're made of repeating DNA sequences. For example, repeated sequence TT, A, for example, and G, 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 and so on and over. So its function is protect the chromosome ends from damage or sticking together. Okay. So its function is to protect with it's the protection of the chromosome ends. You're going to protect their end caps. They're going to protect the chromosome ends from the damage and for the sticking together. If the chromosomes can stick together, right? So it's protecting them from it. So telomeres, they're going to get shorter. It's very important. Telomeres are getting shorter with the age, which is linked to aging and the cancer. Okay, the more you age, the less the telomeres. The length of the telomeres will be also less. The older you are, the less the telomeres. Okay, the younger you are, telomeres will be long. Okay. So shortening of the telomeres is according to your age, which is linked also to the aging and the cancer, okay, in your body. So what about spindle fibers? Let's talk about the spindle fibers now. So spindle fibers, as you can see here, the spindle fibers, they are used during the mitosis or meiosis. These fibers grow from the they are going to grow from the centrosomes and attach to the centromeres, okay? So let me show you the centrosomes here. So as you can see, this is in this cell. In this cell, you can see the centrosome, right? Centrosomes are made of two centrioles. These are centrioles, two centrioles, okay? It's also centriole. So centrosome is made of two centrioles. So they are used during the mitosis and meiosis and attach to the centromeres. They are going to attach to the centromeres. Here you can see the chromatin. Okay, this is centromere. So these are two centrioles. So they're going to make the spindle fibers. They are making the spindle fibers, and each spindle fiber is going to go and they are maybe attached to the centromeres of the chromosomes. Don't confuse the centrosome with centriole and centromere. Three of them are different things, okay? They pull the sister chromatids apart. So they're going to pull one, one centriole is going to pull the one chromatid to one pole. The second centriole is going to pull to another pole, okay? So they pull the sister chromatids apart. This is in the illustration I can show you. These are the spindle fibers. These are the spindle fibers which are made by centrioles. Totally, we call it centrosomes. So you're going to pull the sister chromatids. You see, sister chromatids work together. So they are together now. They are together now. But when it's pulling, it's going to break down the centromere, which is attaching two sister chromatids together. They're going to detach it. So the spindle fibers are going to pull it to the poles. So this is the feature of centrosomes. So think of them like ropes pulling the chromosome halves to the opposite sides of the diploid cell. Okay, so we learned the concepts which we call the telomere shortening. So telomere shortening, telomere is responsible for, this is the end cap which is responsible for your aging. The more age you are, the less telomere will be, the shorter the telomere will be. Okay, so it's related to the aging and the cancer. So telomeres get shorter every time a cell divides. Every time a cell divides, it's going to get shorter, the telomeres. When too short, the cell is going to die or it's going to be mutated. Okay, so sister chromatids, they are Chemotherapy targets many anti-cancer drugs. They stop the chromatids from the separating. Okay, so sister chromatids won't separate them, stopping cancer cell division in this case. Okay, what's centromere? This is centromeres can lead. This is the centromere. It's the belt in the center of the chromosome. This centromere can cause a lot of genetic disorders. For example, incorrect attachment of this centromere can cause missegregation, which can lead to the Down syndrome or which can lead to the Turner syndrome. Okay. We have the chromosome arms, which is P arm and the Q arm. So it can lead to the cystic fibrosis gene. We have on the 7q31 region. In the 7q31, there are some genes which are located on specific chromosome arms. If they are mutated, we call it genetic mutations. So telomere diseases, we have telomere diseases, telomeres. So this keratosis congenita, I'm not going to stop it. So it is inherited disorders due to the telomere maintenance problem. So there will be bone marrow failure, okay? So we talked about the spindle fibers. We talked spindle fibers, they pull chromatids apart during the cell division. They are like spindle fibers, they are fibers which are pulling the chromosomes apart during the cell division. Telomeres, they are the end caps, of the chromosomes. Their function is to protect the chromosome ends. So P arm, Q arm, short and long arms of the chromosomes. We talked about the centromere, which is connecting two sister chromatids together. Sister chromatids, they are identical copies joined together. And the chromatid itself is a half of the duplicated chromosome, right? This is a half of the duplicated chromosome. One sister chromatid, this is half of the duplicated chromosome. And the chromosome itself, this is the coiled DNA, which is ready for the division. I hope you understood everything. From the next lecture, we're going to talk about, we're going to enter to the cell cycle, okay?